Diego.
Okay, so good day, everyone. On behalf of the Interpore Brazilian chapter Pore Talks team, I welcome everyone. Thank you for joining us and supporting the speakers today. This is the third Pore Talks live session. I have the pleasure to co-host this session with Anderson Camargo. And today we have two speakers, Berilo and Daphne. Thank you, Berilo and Daphne, to be here with us today. And at the end of each presentation, we will have a Q&A session. So please feel free to send your questions or comments through the live chat during the talks, or our team will bring them to the speakers when the time is ready for the questions and discussions. And don't forget, enjoy the session. Do you want to support us and the speakers? Hit the like button or leave your kudos in the comments. And without further ado, take your seats and let's get started. I will start presenting Berilo. Berilo is a PhD candidate at LAMAP. LAMAP is a lab in the Federal University of Juiz de Fora here in Brazil, and a guest researcher at TU Delft, and a member of the Porous Media Time Talks team, currently conducting research on foam flow in the context of enhanced oil recovery and geologic carbon sequestration. His work focuses on numerical simulations that investigate the influence of permeability heterogeneities on two-phase foam flow in porous media, and more recently includes lab experiments and simulations on non-isothermal foam flow. So please, Berilo, the live is yours.
Thank you very much for the introduction, for the invitation. It's always very good to meet you all, even if it's online. And I want to congratulate you all for the initiative. It's very nice to see the Porous Media Brazilian community getting more united, more active. So yeah, I'm going to present my work today, as Diego always well said, and his work done with my professors and advisors, Rodrigo Weber dos Santos, Yuri Igreja, Grigori Shapiro, and Bernardo Rocha. I'm going to present the work that is published in the Journal of Gas Science and Engineering last year. The paper is more or less, it is divided like this, but I'm going to more or less follow this structure. So talking a little bit about foam more briefly, if we take a look in this left part of the figure here, we have a gas injection in the porous media. So due to gravitational effects and the heterogeneities of the porous media, we can see gravitational segregation and viscous fingering happening here. And to overcome these contraproducent effects is the application of foam. So instead of injecting a single phase as gas or water, we'll inject a mixture, conjection, or alternating slugs of gas and surfactant solution. So then when we have this mixture inside, the porous media will form foam, which is a dispersion of gas bubbles separated by thin liquid films. Then we'll have the gas trapped in these lamellas formed by the surfactant solution. And then we can, by this, promote a more uniform wavefront and then a more piston-like sweep. In this work particularly, we have focused on the near injection region. So we'll have water, gas, and foam flow. We can represent this in our computational framework as a biphasic flow. And this will give us some insights for injectivity from propagation, gas conformance. We model foam through empirical or implicit modeling. And most studies, even when they assume heterogeneous reservoir, they also consider deterministic permeability fields. So since we have a particular focus on heterogeneities here, we will evaluate the impact of spatial uncertainty. There's a lot of additional challenges for this, but then we'll use some tools to address also the uncertainty related to the geometry of the porous medium. So talking a little bit about the physics behind this, the interplay between rock heterogeneities and the foam flow. The foam behavior will be heavily influenced by the geometry of the porous media. This is a figure of another work of our group from last year. So we have here a permeability field with the high permeability regions represented in red. In dark blue, we have the low perm regions. And here we have the bubble density for this reservoir. As you can see, we have a higher bubble density in the regions that we have high permeability regions here. So there's a lot of stuff related to this. One of the main things, for an example, is that when we have the heterogeneous reservoir like this, we'll have different pressure gradients along our reservoir, and then we'll have different proportions between the foam generation mechanisms. We'll have different values for apparent viscosity observed in this foam mixture in those different parts of the reservoir. And as we have inherent uncertainty related to the porous media geometry, this will also be carried out and propagated into the foam flow and this behavior. So to address this, we'll implement a stochastic framework to explicit account for these permeability uncertainties. We only have a method for discretizing the domain into random fields, and then we can generate samples with controlled and correlated disturbance. So the objectives of the work is perform statistical analysis of the foam performance. We have different foam propagation regimes, and you consider the absolute permeability field as uncertainty input variable. And we can actually quantify the impact of the permeability uncertainty into the foam flow by performing uncertainty quantification and sensitivity analysis. And the framework behind all this is the coupling between Karhunen-Loève expansion and polynomial chaos expansion. So this provides us an efficient and non-intrusive method to analyze this stochastic behavior. We can control the spatial correlation of the samples, and this framework in the context of EOR and foam flow is novel. So a little bit about the models and the methods. The foam flow in porous media. At first, we have a biphasic flow here, water and gas. We assume fully saturated flow. The phases are immiscible, having compressible fluids. We'll have then two-dimensional reservoir here. We have multi-dimensional flow in it. And we consider, as I said multiple times, heterogeneous permeability field, but isotropic, which is also a very common assumption. So the mathematical modeling behind this is done by the governing equation of the two-phase flow. Here we have the mass conservation. Here alpha means the phase when we apply this for both water and gas phases, and also the velocity of Darcy here. The foam model that we use is the CMG stars foam model. Is an implicit approach. So once we have the gas mobility here, we consider the presence of the foam in the domain by altering the gas relative permeability by this mobility reduction factor. Particularly in the stars foam model, this MRF term is represented by this equation, with FM mob being the maximum mobility reduction that foam can assume. And since we have only the dry out effects, consider this the only F function that is considered is the F dry function. And then we have in this function two parameters, the SF bet that represents, corresponds to the sharpness between the transitions of foam quality regimes, low quality and high quality, and SF dry that represents the threshold water saturation that foam will collapse. Therefore, we have MRF. If we have higher MRF, we'll have then a lower gas mobility, and then we will prove sweet efficiency, will delay the breakthrough. For this particular case, we will neglect the effects of capillary pressure, surfactant concentration, and non-Newtonian effects. These are all very common assumptions, mainly in studies like this of uncertainty propagation, because we have to perform many evaluations of the simulator. We must have lots of samples evaluated. Then there's a inherent high computational cost. So a little bit about Karhunen-Loève expansion is a very nice mathematical tool. You can apply with a lot of different goals. In the particular case of our work, we use it as a tool to generate Gaussian random field samples. So if we have, for example, here, permeability, a constant field permeability in one-dimensional case, for example, we can generate as many samples as we want with a random and correlated disturbance. So the core of this tool is this equation right here, where we have this expansion in series, and we can truncate this parametric representation of our space from infinity to this NKL number of terms of the expansion here, and then we can truncate and reduce the dimensionality of our problem. So basically, our permeability field will be represented by this K here, and is defined by a mean value for this field. And each of these terms of the expansion is composed by a random variable, an eigenvalue, and an eigenfunction. And these values here, we can obtain by solving this whole eigenvalue problem right here. We define this covariance function, and then we solve the eigenvalue problem itself to try to make it a little bit more clear and already applied to our case in the bidimensional example, for example. We have our permeability field right here. We start from this baseline, this mean permeability field, which is the 36th layer of the SPE10 project. It's a very well-known benchmark for the scenario of EOR. And then we will build this layering for each number of term of the expansion. We will then evaluate a random variable here in a Gaussian random variable. We'll have associated in this term an eigenvalue and an eigenfunction, and we'll build this layering. For this particular case of this study, we'll have 8 terms in this series. So we build as many samples as we want, but we can then build those samples preserving this characteristic of a well-pronounced preferential path here. But in those different regions of the reservoir, we will have different characteristics. For an example, in this sample here, we have a high permeability region in the lower region of the domain, and a low permeability region right here, the other way around for this other one. And for biphasic flow without foam right here in these animations, as we can see, we still have the preferential path really pronounced here, influencing the flow, but we have different dynamics in the other regions of the domain. And by this, we can address and apply some uncertainty to our permeability field and study the influence of these correlation patterns, global patterns, localized fluctuations, how much each one of them will influence the different quantities of interest that we're going to analyze. And then we'll have insights about these permeability variations. And this all is very demanding, very high computational demand. So we need to apply emulators. So every simulation that we do in this study was performed, and Fossil is an in-house simulator built in our group LAMAP. And for do these uncertain propagation studies, as I said, we need many evaluation models. So when we have the input parameters here, we separate a few of them. We have a direct evaluation in the simulator. And as we build a real response between the input and the output, we can train the emulator and analyze the remainder of the input parameters directly through the emulator. This also can be seen as a machine learning technique. We can train a mathematical model, and we'll, in the end, will substitute the evaluation of a simulator that is really, really high cost by a polynomial. And then we can have the outcomes in a very more quick and cheap way. So the tool to do this is the Polynomial Chaos Expansion, and is represented by this equation right here. And we are looking for those coefficients in each term of this Polynomial Chaos Expansion. And by then we can extract each one of the information that we need as mean, standard deviation, and also Sobol indices for our results. So talking a little bit more about uncertainty quantification and insensitivity analysis, UQ will, through UQ, we'll assess how input uncertainties affect the model response. So we'll have all of our random inputs right here. We can analyze directly through the simulator or the emulator, and then we will have our response here with the mean values, the standard deviation, or percentiles, confidence intervals, and so on. And the assay, sensitivity analysis, will come to complement UQ, and by this we can evaluate the contribution of the input parameters directly to the variations of this quantity of interest that you are looking at. So is more or less like this. We can actually quantify of our input parameters here, how much of the variation of the response are related with which one of them. And we do this by analyzing the first order Sobol indexes represented by this equation here. So going to our results, we explored 3 different cases, 2 different foam formulations and the case without foam. And we have 500 samples evaluated for each of these scenarios. The foam A, represented by those parameters here, are considered based on the work of Valdez. And the usage of PCE here provided us a 60% reduction in the total simulation time compared with the Monte Carlo traditionally. So we have here a baseline simulation for both of 3 scenarios, and here the apparent viscosity and the production for each one of them. We had, as expected, as we have a stronger foam formulation here, as we can see through these parameters, we can build the apparent viscosity curves for each, for every foam quality. And for every foam quality, foam A assumes higher values for apparent viscosity compared to foam B, and also to the non-foam case. So as we have a stronger foam, we can reduce the gas mobility, and then the flow will assume this more piston-like behavior. We will have this front more uniform. We will delay the breakthrough and also have a higher water production, as we can see in all these animations. The results for uncertainty quantifications then, for each of these 3 quantities of interest that we analyze, pressure drop, breakthrough time, and cumulative water production, we had the trend as expected. For the production here, as we can see in this figure, we'll have the stronger foam scenario here by this darker curve, followed by the foam A and the no foam scenario. The means, we have the exact result as we already expected, but the variation of the outcome here, we can see when we have a weak foam formulation here, we increase a little bit this variation. This is due the presence of a new physics here. We are increasing non-linearities in our solver, in our whole problem there. So we have a little bit higher variation, but when we have really, really strong foam, we can reduce a lot those uncertainties, and foam with this shows not only this behavior of enhancing the sweep efficiency, but also reducing the uncertainty related to the porous media geometry. We can observe the same trend for both other two quantities of interest. And the sensitivity analysis is what I think is the most interesting thing about this study. So by this, we can actually quantify the influence of each random variable of the expansion here. Each one of those random variables are associated with a different eigenfunction right here. So we can actually correlate which of those patterns here are the most influent in our outcome. So for pressure drop, for an example, we can see that the first fluctuation mode here carry out the influence in every time step for every case. I put it here for the strong foam one, but for the three scenarios, the results were pretty much the same. So this fluctuation mode here was the most influent, which was already expected because this fluctuation mode is associated with the largest eigenvalue. And this, its shape also provides a higher influence on the mean permeability of the field. So for breakthrough time and production, we have pretty much similar results here. We have the modes 3 and 4 being more influent for, sorry, I clicked here. We have the modes 3 and 4 here being more influent in every time step as well. And for breakthrough time, they carry out most of the influence. This is due this geometric shape that they assume that are more related to the formation of preferential path. And this shows us how these different shapes can be related to global pattern influence or more localized fluctuation, and how each kilo quantity of interest will be influenced by it. So I'm exceeding a lot my time. Sorry for this. So conclusions, we performed this uncertainty quantification sensitivity analysis for the foam flow, considering absolute permeability field as uncertainty. This coupling between the Karhunen-Loève expansion and polynomial chaos expansion enables us to have a tool to generate correlated and random permeability field, while we could reduce the dimensionality of the problem, and we could achieve accurate results and reduce the computational cost simultaneously. We have those three scenarios evaluated, and we can take out from this that the global scale variation, they carried out most of the pressure drop variability regardless of foam presence or the foam formulation that we had. And local variation, they are less sensitive to foam, but they contribute a lot to the flow dynamics and the saturation from propagation. All this emphasize the stabilizing role of foam. And also very nice stuff about all this is this whole framework can be applied into different cases. It doesn't need to be related with foam flow. It can be applied in also different kinds of analysis or physics and so on. And for future work, we pretend to use different foam models with non-Newtonian behavior and this dynamics of generation and destruction of bubbles and dependency of phase velocities and capillary pressure. So thank you all for your attention, for the invitation. Sorry that I exceeded my time. And I open to questions now or at the end of the session. I don't know. Thanks.
Okay, thank you, Berilo. Now we have time for one question.
Okay.
I'm not sure if we have questions in the live chat on the YouTube.
No, in the live chat, no.
Oh, okay. So I have one quick question, more like a curiosity from my side. From the perspective of the recovery production, do you think that the delayed breakthrough time could be considered as a quantity of interest, or as we like to say, QOI, a QOI to devise how we could design an optimal flow? I mean, the delay is probably due to the induced diffusion and increased viscosity of
Yes.
the flow mixture with the fluid, right? Oh, so I'm not sure
Yes.
if you have the perspective to study the fluid characteristics and the mixture characteristics from a thermodynamic point of view, for instance.
Yeah, we are expanding in the more to the thermodynamic point of view now, the work, but they are very related, both QIs, as I presented them both together here, but I think it's more easy to visualize here. For an example, we can see clearly the breakthrough time here when we have this change in the behavior of the production curves. So this is, we can see this through these animations here as well. So as we have the gas mobility changed by a foam, then we can do a better sweep and also leave less water behind the front. So if we have these simulations with fixed velocity and we have all these changes happening, we'll, by consequence, we will have the delay breakthrough. Also, there's all very coupled, but I passed through very quick.
Yeah, I see.
I don't know if I answered the question, but
no, it's fine. It's great. Really interesting work. Congrats to you and for your co-workers. Really interesting and great work.
Thank you very much.
So
Okay,
I
thank
think
you, Berilo. Sorry.
think that sorry, Anders. I will just pass the word to you. So please feel free to conduct from here.
Thank you, Professor Diego. Berilo, very nice presentation. I also have two questions. However, I'm going to send you by email, okay?
Okay.
Okay, if you don't mind. So I'm going to
course.
send you very soon. And let me introduce you all. My dear friend Daphne. How are you, Daphne? Are you fine?
I'm fine, thank you.
Great, Daphne. Daphne Pino is a hydrogeologist with a master's degree in earth science, from the University of Quebec, Canada, and a Ph.D. in hydrogeology by the University of São Paulo. Her research topics include hydraulics of fractured aquifers and hydrochemistry and microbiology of porous aquifers. Nowadays, Daphne is a postdoctoral research at the University of São Paulo, and she is also an invited teacher in the postgraduate subject of hydrogeology in the Geosciences Institute, also in the University of São Paulo. Congratulations on your work, Daphne, and the E-floor is yours, okay?
Thank you everyone. I'd like to first thank the Interpore Brazilian chapter for organizing these talks and for everyone who has joined us through YouTube link. So my topic today is Beyond the Invisible Resource, Assessing Aquifers at a Pore Scale. So we typically hear about aquifers when we're discussing resources at a regional scales, like when we talk about the infamous Guarani Aquifer here in Brazil. However, groundwater is not a visible resource like rivers, so they have been called an invisible resource even by UNESCO for a couple of years now. So, and the risk of this invisibility is that we don't directly see its depletion or its degradation in real time. So it's like the saying goes, out of sight, out of heart. And in that context, pore-scale observations are essential for accurately upscaling flow models and for addressing global freshwater challenges. So the idea today is to show you a couple of examples of pore-scale studies applied to aquifers. So aquifers account for 30% of the available fresh water in the world, and so it's an important resource for addressing current global issues such as world population growth, agricultural intensification, and increased water use in different sectors. The contamination of groundwater, it's a severe threat to public health, to ecosystems, and even to local economies. And however, in the past decades, we had some technological advances that improved aquifer characterization. And these aspects, they are relevant when we're thinking about the United Nations Sustainable Development Goals, particularly number 6, on securing clean water and sanitation. In Brazil, for instance, 52% of the municipalities use groundwater for water supply, and that percentage is even higher when we're talking about the state of São Paulo, where 80% of the municipalities rely on groundwater for water supply. However, water supply is not just a matter of water availability. In the metropolitan region of São Paulo that you see here in the map, we have observed an increased interest in groundwater use, particularly during periods of drought, like what happened 10 years ago in 2014, 2015. And although we have around 14,000 private supply wells for water supply in the metropolitan region, which are the black dots that you see here in this image, all those black dots are supply wells. We see that many of them, they are near contaminated sites, which are the green circles that you see here, or even near industrial areas, which are the orange areas. So there is a chance that many of those supply wells, they are actually extracting water that might be contaminated. And we know that regional or microscale groundwater flow and contaminant transport is ultimately controlled by pore-scale mechanisms. So we're going to see a couple of examples of pore-scale studies that are focusing on groundwater contamination problems. And we're starting with a very common type of contaminant in industrial sites, which are the chlorinated solvents. So these contaminants, they are called organic chlorinated solvents. The most used ones are PCE and TCE that you can see here on the left. Vinyl chloride is here because it is a product of their partial degradation, but is actually the most harmful to human health. And these compounds, they can affect brain, kidneys, liver, and the cardiovascular and reproductive systems. So we definitely don't want them in our groundwater. So we did a study to observe the degradation of TCE that you see here by potassium permanganate. This is actually a very common application in the field, which it's what we call an in situ chemical oxidation, as we oxidize the TCE using potassium permanganate. So what we did an injection experiment using glass beads to simulate the porous aquifer at a microtomography beamline at the synchrotron facility. So we injected the TCE in the saturated sample to mimic a contamination event. Then we flushed it with water because, well, after the contamination, the water in the aquifer keeps flowing. And finally, we did injections of permanganate to react with the remaining TCE. And something that is interesting that we saw is that after a water flush, we see some droplets of TCE that were trapped within the smaller pores or within pore throats. And when we start injecting permanganate, here we have the solution of potassium permanganate, it took a while for it to start consuming the TCE. So we kept injecting permanganate, and after some time of injection, we noticed an increase of the pressure in our system. And when we looked at the images after this pressure increase, we see that a film formation where we had before our droplets of contaminant. And this is very interesting because the shape of these films they are quite unique and they are very typical for fluids. And considering their shape and the increase in pressure in the system, these films, they are interpreted as the precipitation of manganese dioxide, which is a product of the degrade when potassium permanganate reacts with the TCE. This is very important because it can lead to pore clogging and also to changes in permeability, and this is going to affect the remediation process, even when we're doing injections in the field. This is just another example of film formation, where we previously had a drop of TCE, just to show that the other one that I showed you was not a single event. So we had this happening throughout our sample. In another experiment, we were testing the mobility of zero-valent iron nanoparticles, and they're very interesting as well because they are capable of degrading chlorinated solvents, like we see the TCE here again. But in the study, we were not using TCE, we're just looking into nanoparticle mobility. So we did another injection experiment in front of a microtomography beamline as well. And a distinct aspect of this experiment is that we did 2 nanoparticle injections here. And this is different from previous studies because they were just using 1 nanoparticle injection, so they were stopping at step 3 here. And why this is important? Because when we're in the field, when we're doing remediation projects in the field, we are going to inject we're likely going to inject multiple times. So we wanted to see if this has some sort of effect in our sample. So we start with our sample saturation. Then in the first round of nanoparticle injections, we can see they are the lighter shade of gray here. We can see that some areas very bright white here, they had a more concentration of nanoparticles. Other areas had lower concentration. Same a bit uneven distribution. With the second water injection, we flushed most of those nanoparticles away. We can see that most of them went away from our sample. Then with our second nanoparticle injection, we actually were able to have a better distribution of nanoparticles through our sample. We see that it's actually a bit harder to see the grains from the nanoparticles. The grains are the darker gray here. And when we did the final water flush, we did not flush away as much nanoparticles as we had before. We see some clusters, some nuggets of nanoparticles here within pore throats. We can also see this in this graph. So we had our baseline, our porosity here in light blue. Then part of our porosity was occupied by nanoparticles. So we have this reduction of porosity here. These pores that I'm showing are just pores occupied either by air or by water. Then we inject water for the first time. After the first nanoparticle injection, we recovered most of our porosity. When we injected nanoparticles the second time, we were able to inject a lot more nanoparticles here, but when we flushed them out, we had a greater retention of nanoparticle within pore throats and smaller pores, which is what we see here in the dark blue. So what we learned from this experiment is that nanoparticle injection history plays an important role when we're discussing mobility and entrapment of nanoparticles in porous media. This one last study that I want to show you, it's a bit different now. We're talking about nitrate contamination problems that we also have in urban areas. I know we usually hear about nitrate when we're talking about agricultural areas because of the application of fertilizers, but in urban areas, we can have nitrate contamination from sewage systems. And just in the state of São Paulo, we have an estimation that around 10% of the sewage volume is lost and it becomes recharged into our aquifers. I know it's a bit scary. So nitrate is a very common contaminant in urban aquifers worldwide because of its high mobility. And in natural systems, we have a process of nitrification and denitrification that are controlled by bacterial activity. And at a micro scale, these processes, they can occur at the same time because we have the formation of microenvironments with different redox conditions. So if we have the heterogeneity in the distribution of oxygen, organic matter, and water, especially within those small pores. This is going to be fundamental to control these reactions, and this also highlights the importance of using approaches that account for this pore scale variability. And as we spoke of some impacts before, the consumption of water with high nitrate concentration is associated with the baby blue syndrome, which is blood disorder that prevents oxygen to be delivered to body tissues. And in terms of environment, we can also have some ecological problems such as eutrophication. So in this study, what we want to we want to understand the controls of microbiological activity regarding the nitrogen transformations in an aquifer that is affected by a nitrate contamination. So for this, we're using microCT images and thin sections, granulometry analysis, and X-ray diffractions to characterize our pore space geometry. We are also doing DNA sequencing because as the nitrogen cycle is controlled by bacteria, we want to know which are the microorganisms that we have in this aquifer. And we are also planning some small flow-through bioreactors so we can study bug flow and define some denitrification rates in our samples. Okay, so far we have already been able to identify some different types of pore spaces in our samples. Because of time, I'm going to just show, I'm not going to read each one of them, but we have here microtomography images on the left and thin sections on the right. And what we expect is that these different types of porosities, of pore geometries, that they will be relatable to what we see in hydrogeochemistry and to regarding bacterial communities. So we have this, what we got so far from our microtomography analysis. We had some results on porosity, and they have been relatable to numerical models of the plume. This is the plume model for the whole city. And this study in particular, it showed that the nitrate plume, it follows urban expansion and sewage network development in the city. And we have like the numbers that we're finding on our microCT images, they are in accordance with what was used in this model, so we're very happy about that. We also have some hydrochemical data to support our observations. For instance, we have here these 2 points in yellow that they have very low nitrate, so you could tell me, well, we don't have contamination there. But yes, we do, because we have high contents of chloride, so this indicates contamination. So where is this nitrate going? And that's why we're doing these studies. We're currently doing DNA extraction from our samples so we can look into those microorganisms, and hopefully in the near future we're going to have some very nice data coming together very beautifully. So to sum up, these studies we're trying to bridge the gap between what we see in microscopic physics and large-scale prediction in aquifer studies. We can evaluate remediation reactions that can cause pore clogging and mineral precipitation, and those will drastically change what is happening in terms of hydraulic conductivity and contaminant flow, which is essential when we're studying aquifer and aquifer contamination. We can also look into the trapment of substances in pore throats or in stagnant pores. And currently we're doing a study looking to understanding the environments that favor bacterial activity. And ultimately, pore structure heterogeneity may alter contaminant flow and entrapment. And one very important point is that if contaminants are trapped within aquifers, they can stay trapped for decades, and they can slowly diffuse from those stagnant zones long after the main plume has passed. So understanding these mechanisms at pore scale, they are extremely valuable for understanding aquifer processes. And with that, I'd like to express my appreciation for all the funding agencies, researchers, lab staff, and students that were or are still enrolled in the studies that I have shown you today. And if anyone would like to know more about the Nitrate Project, we have a website and we also have social media. Well, thank you very much. I think I just crossed 1 minute, so we might have time for some questions.
Just in time, Daphne.
Okay.
Thank you very much for your presentation. Very nice
Thank
talk.
you.
So I have a first question. You showed us a map with different points, with different places with red dots. I could see some companies there in that map. So I would like to ask you if in every dot, in every place, the contaminants are different from each other, or they are all the same?
The first one in the beginning where we had the supply wells.
Yes, because I believe that you can have more oil in one place, more nitrate in other places. So in every wheel that you showed, I believe that are different contaminants there.
Yeah, we had, oops, sorry.
Right in the beginning.
Oh, in the beginning.
Yes.
This one.
In the groundwater. Yes.
Okay. Yes. So actually, these are different. We have these red ones that are contaminated sites, and we have the black dots that are the supply wells. The supply wells are just for extracting groundwater. It could be for different types of use. And the red dots, they are contaminated sites that are registered in our environmental agency. So here in São Paulo is CETESB. And so they are registered. We know they have a contamination. These are not just for nitrate. This was in the beginning of the presentation. So these are for many different contaminants. Could be for chlorinated solvents, could be for metals, could be for, they could be gas stations. They could be a different range of
So for every kind of contaminators, you need to use a different nanoparticle.
Yeah, not necessarily the nanoparticles will work for every single contaminant that we have here, especially because in many of those sites we have more than one contaminant.
Okay.
The case that I was discussing, I was focusing the nanoparticles on chlorinated solvents, which are solvents used in industries. They could be used for degreasing, for removing, I forgot it. I think it's not grass, from removing oil from parts, from metallic parts. They used to be used in dry cleaning. Now they're banned from that use. But the contamination is already there because in the 50s and the 60s, we didn't have this concern of how are we disposing these solvents, what are we doing with this? So there is a lot of contamination that has been there for decades.
At least the rock is the same. It's sandstone.
Here we have
in São Paulo.
Yeah. This is why it's so much fun when we're presenting groundwater to people that are not.
Okay.
In São Paulo, we actually have two aquifer systems. We have the upper one, which is it's not exactly. Yeah, we have some sandstone. We have sediments, sedimentary rocks on the upper system. But actually these systems, I say upper system, but it can go down to 150 meters in depth. And below that, we have gneisses and schists and granites that which form what we call the crystalline aquifer system. So we have two different systems here. A lot of the supply wells, they are extracting water from the fractured aquifer system because there is usually contamination is going to affect the shallower portions of the aquifer. But when we look for this place here is what we call Jurubatuba. It's historically an industrial area, and they had lots and lots of supply wells in there because it took a while for water supply system to get there. So a lot of industries needed the supply wells. So imagine at every property you had a supply wells extracting water 100 or 150 meters below ground surface. So this is going to pull our water level down, and whatever contamination you have up here is going to be dragged down. And what we see is that in those it's deep. It's not deep in terms of oil, I know, but it's deep in terms of groundwater extraction. If you're extracting 150 meters down, you have contamination there. So we have, and you have different types of contamination here because this was mainly an industrial neighborhood. So now it's changing use, but we still have a lot of contamination problems here. So we don't have a single answer that works for everyone, unfortunately. Otherwise, if someone comes up with that, you're sitting on a gold mine, but it would be brilliant. But yeah.
Okay, thank you. It's a difficult job indeed.
Yeah, it's very nice though.
Okay, so do you have more questions?
Sorry, I get excited. I start talking about very different things.
It's a very nice talk. So if you don't have more questions,
yeah, may I have one?
Yes, go ahead.
A really great talk, Daphne. Regarding the biogeochemistry, I was wondering, do you have the plum simulations, right? So in the plum simulations, do you have some, activity models to simulate the ions flowing through the fluid? Do you have some dedicated model to handle the biogeochemistry reactions, for instance? Or it is just the transport of the nitrate and the contaminants and the chemical species in general?
Okay. This work here, that is the one when I mentioned numerical modeling. This is at a very different scale. This is a city in the state of São Paulo, and it was modeling nitrate contamination for the whole city. So it's a very different scale from when we're looking into our 2-millimeter plug of rock.
Yes.
We are going to simulate flow here, but we also have another person. This is actually a very big project in FAPESP, so we have lots of PhD students working here, and also a couple of postdoc like myself. And we have someone working with the microtomography images and doing simulation flows, because we also did, I didn't have enough time, but also did injection experiments in the main line here. So we're comparing with the simulations. We also have someone modeling, working with the hydrogeochemistry at a more local scale here in the city. But I don't think we have so far a software to simulate the reactions, to simulate bacterial activity. We were discussing this the other day, where we're actually collecting different lines of evidence that is going to show, well, we know this is happening. Like here we say, well, you could say do we have denitrification? Because I don't have a map here, but an interesting point is that we have some of the wells here that I'm showing here, they are near a known source. And then we have some wells that are upstream and wells downstream. And these two, they are downstream and they are shallow wells and they are near the river. So we think there might be something near the river that is favoring the consumption of nitrate here. But we also have other wells that are near them, but they are deeper and they are not, we don't see this consumption of nitrate. So we want to understand what is happening. And in those wells, we have collected soil samples that we're doing microtomography and DNA extraction. So we want to see, is there a very big difference that we see in terms of pore structure that would justify having different environments that would favor different redox conditions for consuming nitrate? Do we have different bacteria that are consuming this nitrate, or do we have a species that is inhibiting the nitrate consuming bacteria, so they are not active in one portion and are in the other? So we're actually going through different lines of evidence, but I don't know. I don't think I have at least I don't have available for using right now a software that would actually simulate this bacterial activity. If you have a recommendation, I'm open for it. Usually people ask me, what do you need? I'm like, depends what we have available. There is a lot of things that we can do in this study.
Yeah, actually,
it would be very neat.
To be honest, I don't know. It was more out of curiosity because I think it is a great problem to biogeochemistry, kinetics and thermodynamics. So I was thinking on, for instance, a frequency or even reactor to take this problem and try to simulate the reactions. But I was just curious to see what you guys are using and. But I know that it is
Yeah,
a different scale, but it's one more
we had
like
had someone using
curiosity.
Freaksy for the geochemistry.
Yeah.
But it was mainly for characterizing like general the types of water and things like that. We also have isotopic data that is not here, so this also helps us know. It also helps to trace this contamination. So we have stable isotopes, so hydrogen and oxygen isotopes in water molecule, and we're also analyzing isotopes in nitrate, so in water. So we have nitrogen and oxygen in nitrate molecule in water. So this also helps us to understand these different reactions of the nitrogen cycle. This is still ongoing. We're still analyzing those, but yeah, it's different. There's still a long way to go in a few things, but yeah, it would be very nice. Part of the bioreactor idea is exactly to see that where we see that we have more nitrogen, when we have more bacteria, then we can simulate flow in those bioreactors and see what are the optimal conditions for the bacteria if we change the amount of nutrients of organic carbon, or if we provide different sources of organic carbon, how will they react, and how this would affect the nitrification rates. I actually have more questions at this point than answers, but I think it's part of the fun.
Yeah, it's research.
Exactly.
So, yeah, thank you, Daphne.
Thank you.
Okay, thank you again, Daphne. Thank you, Berilo. Very nice talks today. So I would like to remember everyone that if you want to meet Daphne and Berilo, you can go to Rio de Janeiro next month. Are you both of you are going there? No? Oh my God. I'm sorry.
I'm not also, unfortunately.
So that's nice that you could present your work here. So, but
Yes.
next year you're going to be in Brazil, Rio de Janeiro, okay?
Yes.
Yes, for sure. So I would like to remember everybody so that we are going to skip next month, okay? Because between 10 and 13 of August, we are going to be present attending the Brazil Interport chapter in CBPF in Rio de Janeiro. I hope to see many familiar in different faces there, okay? And I would like to also say thank you for all my team from the Pore Talks, Munarin, Ingrid, Nagata, Diego, Yara. I'm missing someone?
Thais.
Thais, yes, Thais. And I hope to see you everybody in September. Okay? Thank you guys. Thank you very much.
Cheers.
Thank you.
Cheers. Bye-bye.
Bye bye.
Thank you.